Okay, so the talk today is what's neural and what's not an fMRI. So we're going to start out, your typical MRI scan, you just throw someone in the scanner, they're crying for you to take them out, going, what have you done to me? And, while they're in there, you get a series of brain volumes, just across time. That's fMRI. And you have that series of volumes, and for a single point in that, we get a time series. So in this case, we have a time series that goes like this, at this exact location. So what makes us think that this is neural? Did no one else ask questions? Is it too unusual for this course? So looking at this picture, what you see here, what makes you think this is neural?
There's an onset offset.
Yeah, there's an onset offset. Signal goes up and down. Anything else? I mean, that's pretty much the big one. We know there's something that happened 5 times, and we see the signal go up and down 5 times. That makes us think it's neural. So this is a task. This is, a task. What makes us think this is neural? We're doing a resting state fMRI. So Katie talked a bit about that on Monday. And we do an ICA analysis to get a component, and this is what our component looks like. What makes us think this one is neural? Someone I don't work with know the answer to this question? Yeah, I know. Blobs. They're blobs. The blobs are sort of in gray matter. They're sort of where we expect them to be. If you know your neuroscience, this looks kind of like the default mode network. It feels kind of neuralish to you. How about this one? What makes us think this is neural? I've heard a few laughs, so someone has a comment on this. CSF. It's probably not neural. So, in this one, we see blobs in gray matter. In this one, we see blobs in CSF. So there's some very basic things. You can look at your data and saying, no, I don't think that's neural. But it's always good to remember, even when you see things like this, that doesn't mean this doesn't get out there. So this is one of my favorite publications ever in a Glamour journal, Glamour, where the study was studying romantic attachment when you look at people that you love versus you don't love. And this is the map they produced of where the activity is when this happens. So either Descartes was right and the ventricles are the seat of your soul, or they published a very nice motion artifact. I always look for excuses to put this picture in because it's a really fun one. So going through this, in truth, we don't really know that any specific result is neural or not. But for a well-designed and well-reported study, we can be reasonably confident nowadays. But the key word there is confidence. So where does this confidence come from? And there's 2 types of confidence here. One is confidence for neuroscience as a field. So why does the field as a whole say fMRI is worth our time? And the second type of confidence is for an individual study. So you're looking at a specific result, either yours or you're reading in a paper. Where do you get the confidence to say, I buy that, I don't buy that? So like when we looked at that picture of the activity in the ventricles, a bunch of people who know what the ventricles are said, I don't buy that just by, I don't even know what the study design is, I don't even know anything about it, I don't buy that's legit. So that's, confidence in individual study. And that's pretty much what I'm going to be talking about this whole talk. So where does this confidence in fMRI come from? And for confidence in the field, I'll note that Peter Banatini on his first talk, which I think a lot of the summer students probably weren't here for yet, covered a lot of the history of this confidence in much more detail. The talk is online if anyone wants to view it. So I'm going to be covering sort of what, the 3 things that give us the confidence is a plausible mechanism. We have a mechanism that says why fMRI should work. The results we're getting with fMRI match our understanding of how the brain should work. And the results we see with fMRI, we see with completely distinct tools also. So I'll be covering those things first. And then, for the individual study confidence, I'll have a few focused on task, a few focused on rest, and then, if we have time, a task-based case study. So first of all, the plausibility. So the key element for most fMRI is this finding right here, which is that deoxyhemoglobin is an MRI contrast agent. So if you take a test tube of blood, and the blood has a lot of oxygen, and you image it with a T2 weighted scan, you see the test tube. So the blood looks different than what's outside the test tube, but that's it. If you fill it with deoxygenated hemoglobin, then it's essentially distorting the field around it and changing the signal. And if you do this in a rat that is normally oxidated, you sort of see the tissue. If you do this in a rat that has not much less oxygen in it, then you have the vessels popping out. So the basic idea is behind all of bold fMRI is that we can measure essentially we see a signal change depending on how much deoxygenated hemoglobin is in a given voxel. Now, why does this work? Why does this matter? Because the amount of deoxyhemoglobin at any point of the brain varies based on metabolism. So this is a PET study. We have the amount that cerebral blood flow changes during a task versus rest. We have the amount that cerebral oxygen metabolism changes during a task and rest. And we have essentially the division of those two, which is the oxygen extraction fraction. So what you can see is you actually get more oxygen extracted. Is that more? Am I doing this backwards? So what this means is that you're getting more deoxyhemoglobin in a given area of tissue during a task than not a task. And in some ways this is counterintuitive, and this sort of I think Peter covered this a little more in his talk, but it's sort of the opposite of what one would expect. So you're expecting that your body uses more oxygen, so there's going to be more oxygen, there's going to be less oxygen in the blood when you're doing something. But it turns out that the body overcompensates. So when essentially your body uses, let's say, 5 oxygen, but blood flows and sends in 10 oxygen. So the end result of doing a task is you end up with more oxygen in your blood than when you weren't doing the task. And that's a little bit interesting because that was sort of done separately. These findings were done separately from the original fMRI BOLD studies. So that they did the original fMRI BOLD studies, and the several groups that were doing it simultaneously saw that the BOLD signal was going up during a task, and then they sort of dug into the literature and said, okay, why is it going up? We didn't expect that to happen. So in one way, you get sort of a nice history of progression, but another way, it goes back and forth because you sort of take a finding from somewhere else and find how it works, if that made any sense. So how does this look like in a model of everything right now, of how it fits together? So we start with neural activity. The brain is doing stuff. And that could be local field potentials, action potentials, which are like neuron spiking or ion transport. Lots of stuff is happening. This neural activity causes that vasodilation or vasoconstriction, so that the arterioles and arteries in your brain are expanding or contracting depending on what signals they receive. But depending where they expand and contract, you're going to get more blood flow, changing in blood flow coming in and a changing in blood volume. These interact in some ways too. This neural activity is also using oxygen. So you're reducing oxygen. And in a given voxel, your deoxyhemoglobin concentration is a combination of how much blood you have in that voxel. So if your blood volume increases, you have more blood of all types, and how much deoxyhemoglobin you have in that voxel. How much oxygen was used up. So these two combine to give us bold fMRI, which is primarily because it's sort of what's after metabolism. The changes we're primarily seeing in veins of various sizes. So it could be the very small venules to draining veins, depending on parameters. It's worth noting, I'm not going to focus on much in this talk, but there are other methods that can image cerebral blood volume directly, like vaso, which primarily is focused on the capillaries and by measuring volume changes, or ASL, arterial spin labeling, which is primarily measuring changes in the arteries. And what's nice about these two methods is, as you can see, there's a cleaner line from them to neural activity. So there's not this other factor doesn't play quite as much a role. So they have some very good uses. The minus is they're both noisier data and a little bit more complex to collect. So the practical applications are not quite as wide as BOLD. So people like BOLD because it's really easy to do in a lot of different ways. These two methods have some really powerful studies behind them, but they're a little bit trickier. So next, I'm going to show this same flow chart, but over time, to give you a sense of what this bold response looks like. And I should note that I hand drew this with clip art and weird curves intentionally. This is meant to be messy. This is not meant, if I did this too neat, everyone would think this is like, the ultimate shape, but it's messy. So you have some sort of amount of neural activity for a duration of time. While the neural activity is happening, you're getting oxygen metabolism. So oxygen is obviously being consumed by the neurons. At some point, the neural activity is going to trigger changes in cerebral blood flow and cerebral blood volume. Your deoxyhemoglobin, very likely we're going to see a little bit of a drop when oxygen is being metabolized before these two start changing. But once these two pick up, you're going to be seeing an increase in deoxyhemoglobin. And because deoxyhemoglobin follows bold, we're seeing maybe a very brief drop, which is not always seen. Usually, almost always, you can see this big increase. And then we're going to see it return to baseline and usually go under baseline before recovering. And that's, like I said, that's a very rough shape. It varies a lot. There's a lot of factors in here. So the question is, what drives this? So going back to this, the original figure I had, which you can't read now, but hopefully you vaguely remember. We have neural activity, and neural activity drives, is going to affect oxygen metabolism. Did I lose everything in that? Okay, that's annoying. I lost everything in that clip art, in the figure. So the basic idea here is in the cerebral cortex, you have different types of energy using different parts of neural processes using different type of energies. So this is action potentials. This is postsynaptic receptors. These are resting potentials. One of these is housekeeping, one of these is resting potentials. So what that says is the big thing in cortex is these postsynaptic potentials, which is like LFP. But a nice thing to keep in note is that it's actually different in the cerebellum. So when people talk about cerebellar fMRI, there's a lot of things that could vary. So the ratios of where energy is used is actually going to be quite different in the cerebellum than the cortex. We still see bold responses. We still see the signal following the same general shape, but the underlying neural activity might be different. So in this figure, I'm focusing on here, but the other key element here is this vasodilation. Yes? Oh, absolutely.
Would there be a difference in the cerebellum because of the Purkinje cells? Like, is it particular to the cells, or is it particular to the arrangement? Or
both. The big thing is cell densities are very different. So like how the cells, like the amount of neurons versus other cells is different in the cerebellum, and the way the cells interact with each other is also different.
So are these proportions just specific to the neurons that we typically associate in gray matter? Like not with microglia, then maybe it would be different, or do we know?
I don't know off the top of my head. There's a bunch of paper. This is a pretty good summary of what's known in this area from 2012. And they cover sort of a good bit of the different ways people have calculated energy usage, which is a really messy thing to calculate because you have to sort of, in some way, you have to make some estimate of what's using energy and how. And there's a lot of studies that have gone into that. I don't quite know the difference between cerebellum and cortex in energy usage, but the big picture is that it is different, and cerebellum is quite different from the cortex in a lot of ways. So the other part of that was the neurovascular coupling. And this is from a review paper from just a couple of years ago. And I really like this figure for two reasons. One is it shows that we actually do know a lot about how neurovascular coupling works nowadays. And it's not directly driven by oxygen or energy. So there's a bunch of earlier papers in the field, let's say from the mid-90s to the mid-2000s, which are arguing are the veins dilating or constricting based on how much oxygen versus how much glucose? And it turns out that is extremely simplistic. The whole thing that causes veins to constrict or expand is not directly driven by oxygen, and it's indirectly driven by energy needs. So there's a lot of different mechanisms that are defining whether a given vein expands or contracts. So you have astrocytes, which connect to the smooth muscle cells, which are what essentially squeezes the vessels or relaxes them. And there's a bunch of chemical pathways that deal with that. There is the pericyte cells, which have mechanisms that directly affect the endothelium of the vessel walls. You have neurons having mechanisms that also directly affect the endothelial walls. And you have propagation across vessels. So if one part of the vessel expands, it sends signals to other part of the vessels to expand. So there's a lot of different mechanisms. So what this means for the field is there's no one story. So we can't just say we've seen the flow go up, therefore it must be that this is happening in the cells, because a bunch of these different factors can be involved. And how these factors interact is mostly done in animal research. And there's a lot of phenomenal studies out there. Elizabeth Hillman has done a lot of them, Anna DeVore, David Boas. There's a lot of people who have done a lot of really good work on trying to pull apart this model. The other thing I love about this figure, which might not be obvious from the small print, is there's a lot of question marks on it. So for all we know, there's a lot of these mechanisms are like, yeah, we know something's happening here, we're just not quite sure what it is. Something's happening here, we're not quite sure what it is. So it says that this is still a really open field of research, understanding quite what happens in the brain that causes vessels to expand or contract. So one example of this complexity is there's a lot of debate in the field over time on why the signal goes down after a stimulus. So you have a visual stimulation, it goes from here to here, and then the signal goes up during the stimulation, at some point it drops below the baseline and returns. So one theory is that your veins are like balloons that sort of expand out, but then, which brings more deoxyhemoglobin into them, but then they take a while to contract again. And that time when there's essentially more blood and more of that blood is deoxygenated will keep the signal down for an extra period of time. This study is actually saying that's not the case. This study is saying if you do breath hold, we're seeing, so breath hold causes hypercapnia, which is there's extra CO2 in your blood, which causes a blood flow increase everywhere in your brain. Your heart rate increases, it changes blood flow everywhere. So your bold signal goes up during the breath hold and then goes down and returns to a baseline at some point. But the blood volume, so this is using a vasotype measure, also goes up and returns down without going under baseline. And in this case, for the visual stimulation in the exact same subjects, your bold goes up, has an undershoot and goes back, but your blood volume goes up and still returns to baseline. So what this study is claiming is that this undershoot isn't a blood volume effect. They're saying if it's not blood volume, it's not vasodilation, it has to be metabolism. So they're saying that there's continued metabolism use after the stimulation ends. So that gives you an idea of sort of a type of argument that's going on. I don't think this specific paper is the final word on this topic. Very, very likely because of this, that there isn't one mechanism, there isn't one model, but it is a factor out there. I'll also note I've done a lot of stuff on breath holding, and my bold responses actually do have a slightly different shape than this. So even this simple task that we think we know well, there's a lot of uncertainty in it that people are still trying to figure out. So going on to other reasons why we think fMRI is normal. Like I said at the beginning, the results match our understanding of brain function. And this is shown out by the sort of the history of what things were studied. So the first papers observing MRI, observing bold effect in 1991, 1992, and they really started out with very primary things. You move your hand and you see motor cortex signal changing. You show a flashing light and you're seeing an image changing there. And early on, people tried to sort of say, okay, let's do some basic cognition, but most of the work was in these really basic systems that neuroscientists really, really, really knew well what they'd expect. They knew what they'd expect in visual cortex, they knew what they'd expect in motor cortex on a basic level. So early on, the field was really focused on essentially showing findings from decades of animal research in humans with fMRI. And as people gained more confidence, then you started seeing more and more of the basic cognition. You started seeing more and more of the higher order cognition and really squishy things like emotion where people didn't have any clue what to expect because you're not studying emotion in the same way in animals. So that's one example of just sort of how the field changed, gained confidence that fMRI measures something meaningful. And you can see this from the earliest papers. I'm not showing the newest papers, I'm showing some of the oldest. So way back in 1994, someone did basic retinotopy. So they said, if I show a flashing checkerboard on the lower visual field in primary visual cortex, I see the biggest signal on top. If I show it on the top of the visual field, I see it in the bottom. Diagonal, it's the opposite side. So this is retinotopy. This is the retinotopic mapping of primary visual cortex in humans, which has been known since about 1920. But most of those studies were invasive or post-injury. So fMRI was now able to show that the things that we knew with high confidence existed non-invasively in healthy volunteers. And just to show you, then it starts with this, but over time it's gotten, our confidence in these types of findings has gotten really high. So here was a study, and this wasn't the point of the study, but they did it because they could. They said, we know this is, if we project a shape like this, if we put this image in someone's visual field and we stretch on the cortical surface, it will look like an M. And they just used this as their stimuli for the fun of it. And they showed that the warped M that they're presenting to someone's eyes appears like an actual M laid out on the primary visual cortex. So our confidence that we got this good is really kind of amazing. For what it's worth, this is a group at Mass General Hospital, and they used an M, a G, and an H. Similarly, this is going back to the really early days of 1994. People were saying, we know that your signal should change, your response magnitude should change as you increase the frequency of stimulation. So you're making one sound, you're making a sound every 4 seconds, a sound every half second, a sound every 1 second, and you can see that the magnitude scales, which is kind of what we know from decades of neuroscience before that. And then you were showing things that matched in patients. So the WADA test is a common test during certain types of neurosurgery, before certain types of neurosurgery, where they essentially shut down half of a person's brain to see if it shuts down language, and then they know that that's the half of your brain that has language. And it's an invasive, it's an unpleasant test, but it's a pretty decent way of saying, if we shut down the left half of your brain and you stop speaking, that means language is probably important in your left half of the brain. And then when they do neurosurgery in the left half of your brain, they're extra cautious. So what this is showing is in patients where the WADA test showed their language was left dominant, they did a language task, and they find activity in the left. In patients where they found language is right dominant, they found language on the right side. So this is, again, an early study showing that it's matching existing knowledge. For resting state, this was a study that did another interesting population, which was agenesis of the corpus callosum. So the corpus callosum are the fibers that connect the two halves of your brain. And in theory, so actually take a step back. I forget if Katie covered this on Monday, but one of the big concerns with resting state was your vasculature is symmetric. So if you put a seed voxel here and you want to see what's correlated and you see over there, you could say, your two auditory cortices are firing or communicating with each other, or you're saying, no, that's the exact same part of the vascular tree on both sides of the brain, and they're just doing the same thing. So the question is, how can you distinguish those two competing ideas? So what this group did is they said, here are patients who have those fibers that communicate between the two halves of the brain. They never formed. So their left and right auditory cortices are not communicating with each other. When they do an active listening task, task on, task off, task on, both auditory cortices are functioning as normal in these three patients. So both ears work, both auditory cortices work. But if they seed on one side, it doesn't fluctuate with the other, and they seed on the other side, it doesn't fluctuate with the first. So this is showing that, in resting state at least, that this connectivity you see in healthy volunteers disappears when you have those fibers gone, which adds evidence that these fibers actually are what we're seeing, the communication between sides. The next area of this is complementary modalities. So this is, since BOLD happened, what studies have gained confidence that it works. So this was probably one of the turning point studies headed by Nico Logothetis back in 2001, where through a real tour de force of engineering, they simultaneously were sticking electrodes into primate brains and recording BOLD at the same time. So you can see the electrode is here, and they're recording different types of electrical signals right at that point with that bulb activation. And they changed the contrast of the image, which changes the amplitude. And what you could see is with increased contrast, as we knew, bold goes up, and also local field potentials and multi-unit activity go up. And if you sort of plot these things as a scatter plot of local field or multi-unit activity versus bold, you can see it's a line. So when either of those go up, you're seeing this thing go up. And I think this paper was, without question, a turning point in the field, because before that point, any electrophysiologist was saying, sure, you're measuring this relative change in oxygen, but that's not spiking. That's not neurons. We want to stick our electrodes in, and we're getting something totally different. You're never going to get what we're doing. And this paper that did it simultaneously and said, no, actually, it's matching it quite well. Suddenly, that sort of line of questioning became much less powerful. And that was sort of, I think that was one of the big turning points where people really started saying, okay, we can't quite ignore this squishy MRI, functional MRI thing anymore. One other quirk of this paper, if you ever come across it, a lot of people said that this paper said LFPs dominate, so local field potentials dominate the relationship between bold. And if you dig into the paper a lot, you'll see that LFPs model bold better than spiking or multi-unit activity, but not much better. So they're both really close. So sometimes you'll still see people say, like, bold is actually local field potentials, it's not spiking. The answer is it's both, but if you want to sort of push it one way or the other, it's probably a little bit more LFP. Again, there's other methods. This is optical imaging, which is essentially shining different frequencies of light into tissue. This example comes from rat. And what we're seeing here is we have the total hemoglobin, we have the oxyhemoglobin and the deoxyhemoglobin. And over time of stimulation, you can see that the total hemoglobin has one pattern, the oxyhemoglobin has another problem, the deoxyhemoglobin has a different spatial map. And these maps actually match pretty well what we're seeing in bold. So this is a totally different measure. It's a direct measurement of specific vessels, and it matches what we're seeing with bold. Back to humans, there's been a lot of work on simultaneous fMRI and EEG. This is one of the first papers to have done it, and what you can see in this paper is that alpha, so low frequency EEG, as that band fluctuated, we saw one network of areas that was anti-correlated to the alpha fluctuations, another set of areas that it was positively correlated to the beta fluctuations. And I will note that there's a lot more papers and a lot more detailed papers on EEG fMRI. Katie talked about a bunch on Monday. But this is just showing, like, as the field is building confidence, this is one of the papers that sort of aided that confidence. And like I said, this is a lot messier. So here's a study that showed you that alpha frequency that we were just talking about. It's showing that fluctuations in alpha over time actually vary with fluctuations in your breathing depth over time. So if you're not sure if your signal changes are breathing or alpha, it turns out that those two are in sync, which, if you know anything about EEG, kind of makes sense, because alpha is sort of an attention, a rough marker of sort of attention and awareness. And your breathing rate might change depending on how focused you're on something. So this is a confound possibly for other things too. The last thing in sort of this field area is, a lot of the electrophysiology stuff, people are always talking about fast signals. So they're talking about, like, you stimulate something for 50 milliseconds, you see your neurons fire, your neurons stop firing within 100 milliseconds after that's done. And usually with bold MRI, we're measuring things on every 2 seconds or so. Now things are getting faster, but the hemodynamics are slow. So the hemodynamics are not going to be faster than 2 or 5 seconds or so. So what are we measuring these slow 5-second drifts when everyone knows that neurons are firing really fast and recovering really fast? So what this study is showing is that if you actually look at the whole spectrum, so these are different frequencies, and you look at the correlations between areas, there's actually a lot of power in these low frequencies. So even if there's a lot of stuff happening at a high frequency, these areas are fluctuating over time at fairly low frequencies. And I like this for two reasons. One, it's a really cool study. Two, this is the type of questions that fMRI informed the rest of the literature. So this is the type of thing that before fMRI started seeing all these really slow things, a lot of the electrophysiology world wasn't really looking at what happened at slow speeds because electrical activity is fast. But once fMRI started seeing these slow things, a lot of people started looking in the electrical signals at slower things, and it's been quite a fruitful area of research in a lot of ways. So I'm going to switch to the second part on sort of how do we get confidence in individual studies. Before that, I've been journaling on for a while. Any questions? Okay, I have explained everything perfectly. That always feels great. So going back to the initial overview, where does this confidence come from? So I'm going to be talking about for individual studies, a little bit on task, a little bit on rest, and then a case study. So I'm going to go back to the original thing that I showed at the very beginning. How do we know this is neural? So at the beginning, we said, you know, this 5 seconds block, signal goes up, down, up, down, up, down, up. That looks pretty neural to me. So that's where we started. Here's another voxel in the brain. Here's another voxel in the brain. Here's another voxel. Here's another voxel. So what do people think? Do people still think this is neural? Yes?
I mean, you have to know if they're moving too. Important. Like if they're moving with each block, then that would be an issue.
So while you're thinking of motion now, then so you said I'm going to repeat questions just because I'm recording. So you're saying that there might be movement. So why are you thinking that there's a movement issue here and you weren't thinking about it a movement issue before?
Oh, I walked in late.
Oh, okay. Okay.
Yeah.
So the answer was he walked in late. So why do you think so people who are at the beginning, why everyone was pretty confident this was neural at the beginning. Why do you think motion might be crossing people's minds now? What do we know now that you didn't know before? Yeah.
The neuronal signal we're looking at varies over these 5-second times, like the bulk signals varying over that time. And those time scales are time scales where motion, repetitive motion, could happen.
Okay, so that might be one of the motion. And Ben says thing else that jumped out we didn't know before, that we have the same signal going up and down, but it's happening across the whole brain in pretty similar ways. So suddenly when you're seeing that, it's like, okay, what neural task would be showing signal in all these different areas and in our CSF again? The answer is that type of task is called a breath holding task, which I already brought up. And this is a task that when you hold your breath, you cause a global blood flow increase. So, and it changes blood flow everywhere. And what that means is this probably isn't neural. So this is one thing. So if you sort of looked at your one area of interest and saw a signal, you might say, yeah, I got my effect. But if you started searching around the brain and examining more carefully, you might say, okay, this doesn't quite look like I'm expecting. And just as sort of a side note on that, there's a lot of breathing changes a lot. Actually, I'll actually have a slide a bit later on that. So now, how about the other one I showed at the very beginning? So we talked about how we knew this one is neural. And again, we were saying, it looks like a network that people are familiar with. It's in gray matter. In truth, this map is from motion correlated noise. So this was a fun study that Molly Bright and Kevin Murphy published last year, where they were doing a standard analysis. They're taking their tasks, and they're regressing out motion, they're regressing out respiration, they're regressing out cardiac, and then they're running an ICA analysis to get components. But instead of running that ICA and if they run that ICA analysis on the, on the clean data, so that's the stuff that you normally run it on, we see our nice default mode network. If you're running it on your motion parameters, which is this image over here, you're seeing the same network. So it's a little bit concerning that you see your network on the stuff that you're pulling out of your data. So this map is probably not neural, although I should note one of the quirks is that some true signal is, no matter what you're regressing out of your data, there's no perfect regressor that says, okay, I'm pulling out exactly my noise and nothing else. So some of the things you're going to pull out are going to be correlated to the task. And when you do an analysis like ICA, it's going to be hypersensitive to pulling out exactly what you're looking for. And you know exactly what's distinct. So even if this network is just a really small part of the variance in these motion regressors, ICA is going to be very tuned to pull it out. But it just gives you a word of warning, both about being confident about these findings, also about ICA. Because what it says is ICA is not really a value judgment. It's just a mathematical equation, and it's going to find things like this in the math no matter what. And that doesn't mean it's bad. It just means you have to be cautious. So with isolating the neural signal, when we're thinking of how we do our analysis, let's say we have 3 original time series. I have these 2 time series that are perfectly correlated. They're identical. They go up and down at the exact same rate. And I have these 2 time series that are really different. They're r equals 0.05, almost not correlated at all. So these are sort of our simulated true data. We're adding in just random thermal noise, scanner noise. Then I'm adding in cardiac and respiration noise, which are going to make everything correlated a bit more. And then let's say they move their head at some point. And it just so happens that that head spike is going to appear in the same direction in these 2 and in the opposite direction of these 2. So let's say you're moving your head to the left, in one direction, then the areas where the brain goes from the edge of your brain slightly in might move in the same direction. On the opposite side of your head, where the signal goes from the same side of the brain, from inside the brain to outside the brain, it's going to go in the opposite direction. So it flips, and what you can see is now after this, these 2 time series that were highly correlated are still highly correlated, but not quite as much. And these 2 time series that were not correlated at all, that's now significant. So when we're talking about removing noise and trying to figure out what's neural or not, this is sort of the space people are playing with. So how do you deal with scanner noise? Maybe collect more data. How do you account for possible respiratory variation? And how do you account for motion? And I'm sort of doing a survey of a lot of these. Some of these have been covered in more detail in previous talks, and they'll be covered in more detail in future talks. But here I'm going to focus on what to worry about, as opposed to a bit more than what to fix. So we got these non-neural, partially bold fluctuations. So respiration and cardiac are actually changing your oxyhemoglobin, your deoxyhemoglobin. There's things like head motion. There's bad task design, which I'm not covering at all, but it could be a big issue in understanding why things aren't neural. There's understanding the effects of collection, understanding the effects of processing. So this is what I was sort of hinting at before with respiration. So here's a study that said this area of the brain we know is really well vascularized. And in this task, they did a memory retrieval task. And sometimes they told the subjects to hold their breath during the encoding part, and sometimes they told them just to breathe normally. And what you could see is in one area of the brain, that didn't really affect the result at all. But another area of the brain, that made a radically different effect. So this says that if you have populations that might be more or less likely to spontaneously hold their breath, which does happen, you got to be very conscious of this. As an anecdote, one of my non-papers for my PhD, and you'll understand why it was a non-paper, is I was trying to study how the effect of stimulus duration on the bold signal. So I was showing several different stimulus durations across several different populations, and I discovered that a non-trivial and I was just showing out flashing checkerboard, and I discovered that a non-trivial number of my subjects held their breath whenever the flashing checkerboard came on. So if I put a checkerboard on for 3 seconds, they'd hold their breath for 3 seconds. If I put a checkerboard on for 10 seconds, they held their breath for 10 seconds. So fortunately or unfortunately for me, depending on your perspective, I collected respiration traces. And I saw that in my respiration traces, and I said, I can't use this data because I can't separate stimulus duration with breath holding. If I didn't correct those respiration traces, I would have been blissfully ignorant and got another publication that would have been wrong, but it would have been another publication. So depending on your level of ethics, I strongly urge you to do things like collect respiration traces and look at them. Again, with head motion, here's a fun study from a few years ago, which there was a group of 100 subjects, and they simply were doing resting state networks. They divided the 100 subjects into 10 groups of 10 volunteers each, sorted by how much their head moved during the run. And what they're showing here is that there is actually a significant difference. If you're just dividing, these aren't different ages, these are sort of a similar population. If you just say what's the network difference between the volunteers whose heads move the least versus the most, you get one map. If you look at the ones closest, you get another map. So essentially, depending on how much your subject's head motions, you can get systematic biases. This is a very big deal because in a lot of clinical studies, particularly clinical resting state studies, you're examining populations that have different amounts of head motion. So if you're studying typically developing kids versus kids with ADHD, chances are your ADHD population is going to move their heads a little bit more than the typically developing. They're all kids, they're all going to move their heads a good bit, but there's going to be a bias, and there's a lot of work in trying to figure out how to deal with that bias. Similarly, in understanding what's neural data, how you collect your data matters. So this is just one quick example. Here is a spatial resolution question. So you're using really big you're using sort of very smooth data versus all the way up to the original data, which is a very high resolution. And what you could see in this case, they were trying to map cortical columns in this case, that the columns are very visible in the high resolution, but once you get to big data, you see a big blur. So here is something where we know this is ground truth, and this is just smoothed out ground truth, but you're losing that if you're actually just collecting this type of data or this type of data, you're losing that information. So this is going to affect on both what you consider neural and how you interpret the data. Data processing matters too. For example, there's a still fairly common but controversial pre-processing step in resting state where you regress out the global average of the entire brain before doing analysis. And the reason people do this is because things like respiration and cardiac signal are going to vary globally. So if you take the whole brain average, you're getting sort of a rough measure of those signals, you're pulling it out, and your data becomes cleaner, your results become crisper, they become very beautiful. They look like this. Nice, very crisp, nice, clean areas. Unfortunately, essentially by the pure math, when you're taking a bunch of correlations and you're regressing out the global average, you will always, always, always get anti-correlated signal. So that means some areas, whatever your seed is, you're going to find some things that are positively correlated to the seed and always find things negatively correlated to your seed. So by doing this analysis step, you're essentially distorting the results. This doesn't say there isn't such a thing as anti-correlated networks or that they don't exist. It just says that if you're doing this step, going back to the question of this talk of what's neural and what is not, by doing this step, you lose the ability to truly understand, to truly get at the level of the data and understand the differences. And more importantly, just taking this beyond the global signal debate, and it's been a fun debate for the people that are sucked into it over the years. I think the broad take-home message is if you're removing an uncharacterized signal, it's going to cause uncharacterized changes in your data. And that becomes really a big deal with population studies. So if, there's been a bunch of studies now showing that the global signal actually has some systematic differences across populations, which means if you're regressing it out, you're systematically changing what your resting correlations look like. And what I recommend for that, if you want to account for this, if you have any steps you don't do, what I often do is I look at the correlation maps of my noise time series. So like if you're regressing out something like global signal or CSF or white matter, you can actually look at the correlation map of your CSF signal to the whole brain. And if there is a population bias in that, that's a big red warning flag of what you're doing next. So in that case, it's a nice thing you can actually at least correct, at least gain more awareness of if it's a problem in your specific data set to give you more confidence if it's neural. Also, modeling response shape can matter. Here's in this study, in 20 volunteers, we just looked at a flesh and checkerboard and had people move their hands very briefly. It was just like a one motion. And we said, how does the hemodynamic response look in motor cortex? And what you can see is it varies quite a bit. Some people peak at 4 seconds. Some people have the signal peak at 6 seconds. Some people have almost no undershoots. Some people have gigantic undershoots. So there's a lot of variability. And the irony of this paper is a lot of people sort of left it like there's a lot of variability, and it's most frequently cited by people saying, I didn't get the finding I wanted. Maybe it's just because we can't model hemodynamic variability. So it's often used as sort of an excuse citation, which is frustrating, although since I'm the author, I don't mind the citations. But there's a second step of it, which sort of shows what this means. So it says, what I'm showing here is if you're trying to take these magnitude estimates into a second level analysis, you can see if your peak is a second off, you're adding about 10% noise to your second percent analysis. So anything you do to sort of get that better, a better estimate in your individual subjects is going to remove a lot of noise in your higher level group analyses. Another thing there's a lot of work over the years on sort of modeling the order of neural events, such as you want to say, in some sort of study, brain region A is driving the signal in brain region B. And a lot of people will give you different messages. My extreme bias on this topic is anything that does this is really dicey. Because not only does the hemodynamic response vary across individuals, it varies across brain regions. So if you have a larger vein, the hemodynamic response is going to be different than an area with just capillaries. If it's a different area of the vascular tree, it might be happening. If the big neurovascular coupling slide from the beginning shows all these mechanisms, there's a lot of things that can vary the hemodynamic response shape. So if you're trying to say that because a hemodynamic response peaks earlier in one location than another, you're almost always on very shaky ground unless you can have some sort of internal control in a study, which I could possibly count on one hand, if I can count on one hand, the number of studies that have done that. So this is an example of a simulation using dynamic causal modeling, which is sort of one method that does this. And what I did here was I took two simulated time series with the exact same hemodynamic responses. Everything was perfectly identical. And I just added noise, and I said, does DCM say one node predicts the other? And does node 2 predict node 1? It's below this significance threshold. So no. Does node 1 predict node 2? Does node 2 predict node 1? They're below the significance. By the two colors are just different levels of noise. And then I can say, if I give it no data at all, same thing. It's not saying there's any prediction one way or the other. So these two give me nice confidence. Then I'm shifting the peak or the onset by 1 second. And now it's saying that one of the nodes predicts the other nodes. In some ways, that's good because that's exactly what this method should be doing. In some ways, as is known, a peak shift does not necessarily mean it's modeling the underlying neural signal. The most disconcerting one here is this one. So here what I'm doing is in one of these two time series, I'm just making the undershoot. So the signal peaks and then it goes under baseline. I'm making that undershoot bigger. And now, so that the peak is happening at the exact same point, the stimulation time series is having the exact same point. Just saying that the undershoot is bigger in one subject says that node 2 predicts node 1 at a really high rate. So this is an example of this really complex advanced mathematical system doesn't model hemodynamic variation at all, and that variation can significantly bias the results. I've talked a little bit about population biases. Here is an example where this is a study of cross populations using just seeing if there's a difference between younger volunteers and older volunteers. And what we could see is during this task where people were moving their eyes, pressing a button, and when they see a flashing checkerboard and pressing a button, the response in the younger population was almost always larger. When I did that breath holding task, which is sort of a measure of global vascular activity, we could see there were also some variations between younger and older. And when I divided by it, we're still seeing the same effect, but it's not quite as significant. So it's saying that some of this variation we're seeing in the young versus old might be explained by vascular differences between the younger and older population. And we only know that because we collected this global measure. So I've been talking for a while. I have a few slides in multi-echo fMRI, which I think we're getting an entire talk on, which is another way to sort of gain confidence. So I'm going to skip these last few to get to the last little section. Sorry. Allergy season has lasted long this year for me. So the last thing I want to end on is a case study. And this is a fun study where a lot of times we see an fMRI study, you're doing a task for 5 minutes or 10 minutes or 20 minutes, and we see an area of activation. And it's been known for a while that if you collect a bit more data, your area of activation might get bigger. So maybe it'll be a little bit, more voxels will be significantly active if you collect another 10 minutes of data. So the point of this study was to see where does that end. So instead of collecting 5 minutes or 10 minutes of data, we collected 9 hours of data. So this was done over about 9 different days. The exact same 5-minute flash and checkerboard task was done for 9 hours in each volunteer. As I always say, thank you for our amazing volunteers. Not only did they do it, yeah, it was really amazing. And what we found was that as you collect more and more data, the percent of voxels in the brain that become active just keeps going up and up and up without a plateau. So there is no asymptote. If you collect enough data, you will get the entire brain significantly active. And just to sanity check, this is a jar of oil that we put in there, and essentially simulated the exact same analysis on a jar of oil, and that doesn't happen. This is what's called a reviewer request. Yes. So essentially, we did this. They said, is maybe just as you add more data, something is inherently something in your statistics is saying more data is increasing degrees of freedom and everything becomes significant. So we took a bunch of scans with an oil jar and said, no, that's actually not the case. So there's 2 big findings in this paper. One is that more data means there's more significant activation. The second big finding is that the response shapes vary across the brain. So this is a block design. This, classic bold response is what you expect most. We clustered across the whole brain and showed that there are spatially contiguous clusters that have different shapes. So sometimes instead of going up and down, it goes down and up. Sometimes we get these little transients. Sometimes we get only a transient. So here's it's a transient, then it's an undershoot, then it's a transient again. Sometimes it's flipped. It's all over. So the 2 big findings are that it varies, and when we look at the whole brain, there's a lot of cool stuff happening that we're ignoring, that we only get to see when we have 9 hours of data per volunteer. So here's where this becomes a case study. So we got our initial findings. We looked at them. How did we both convince ourselves that this is real, and how did we convince the rest of the field that this is real? So if you saw these figures I showed you, what things might explain these findings that would say that no, this is not neural, this is something else going on? Yes?
So like how you're talking about how increased blood flow in one area of the vessel can influence blood flow in another area of the vessel, where you might end up getting a signal somewhere that there's not actually a neural signal happening.
Excellent. So there's a lot of stuff. One of the classic terms in the field is blood steal also. So, you're obviously going to have in your local area, you're going to have, just to repeat the question, the answer was that vascular signals might propagate over time. So you might have some areas that, the signal just propagates into a wider area of vessels than they started out. And also you might see things like if blood flow is increasing somewhere, it has to decrease somewhere else. So this is a theory that's been around for a while, commonly called blood steal, that you just can't keep increasing blood flow to your entire brain. The strict interpretation of that is actually wrong. So it's the amount of blood going into the brain can vary over time. It's not like sort of a completely closed system. So other thoughts?
Are the patterns just isolated to gray matter? Like it looks like there are some laws there that might show patterns present in CSF.
Okay, so that's another great answer. Is this just restricted to gray matter, or is it white matter or CSF too? So if we're seeing CSF responses or white matter responses, that could be a problem. So that's another good answer. Anything else? Oh, time of day could be a factor. Anything else? Okay. So the list I started out with is luck. We got a few lucky subjects. It's just random this happened. Specific decisions we made in the analysis that bias us toward these results. There's head motion. There's the comment on CSF and white matter. Part of that is voxel size. So if we're seeing things split in, maybe these are like almost 4 millimeter voxels. Maybe sort of the signal is just looks so big because the voxels are so big, and this blood-steal dynamics. For time of day, I will say that most of these subjects were collected at the scans were collected at the exact same time of day per volunteer, and a third version of the study we're now doing, it just so happened that for each of the 2 volunteers, we collected at the exact same day of the week and time of day for both subjects for all 9 sessions, and these findings are still there. Yes?
Were all subjects male or are there also female?
So in this original study, it was two and one. I forget which order it is, and I think all the studies that were the first 2. There are 2 studies that did 3 volunteers each, and both of them were 2 and 1. And the third study is doing 2 volunteers, and it's 1 male, 1 female.
Should it collect menstrual cycle phase for the women?
That is it. So the comment was collect menstrual cycle phase. That is a very good comment. There's been a bunch of research showing that does affect quite a few results. There's some work that's been done here. I think in Karen Berman's group. There's one of my grad school colleagues, Emily Jacobs, has also done some work in this area. So it has been shown to vary, and it's one of those things on the list that everyone says we should be collecting really regularly. It's challenging to collect from a logistic standpoint, which is the frustrating part. I don't think it's an issue for this. It could be an issue for this specific study, both because it's male and female, and because I wouldn't expect an increase based on that. But maybe if we saw response changes over time, or if we, in these 9 sessions, if in these 9 sessions we collect menstrual cycle information, we could have actually found a systematic variation over the 9 sessions because of that.
You should also make sure, or at least document what kind of birth control.
Yes,
you're using.
that is documented.
Awesome.
Yes?
I wonder if mind wandering might also have something to do with it too,
Mhm.
like the complexity of mind wandering over like the 9 sessions,
Mhm.
like what different things people thought about could have
Mhm.
highlighted different areas.
Okay. So actually, I'm repeating questions, so I just wanted to mention that the other comment was record birth control, which I answered, and then the new question is about documenting mind wandering. So that is definitely an issue. For this specific study, there was actually an attention component. So it was a flashing checkerboard, and that was for 20 seconds. And in every one of those 20-second periods, 4 symbols appeared randomly, and they were either a letter or a number, and the person had to press a key for a letter, key for a number. So it turned out that these volunteers, like I said, these are phenomenal volunteers, were almost perfect at getting this task right. So pretty much in every block and every run, they were getting it. So their minds are wandering. There's no question, but their eyes were open. They were looking at fixation pretty much the entire time. In the newest study that I've been involved in, I just happen to know the numbers. They were I think on average, they got those letters and numbers correct 95% of the time, and I think the worst case was like 80%? 75%. So mind wandering is definitely something we were thinking about that I forgot I didn't put on the slide, and that's how we tested for it.
Yeah.
Yes?
Did you find anything interesting in the cerebellum with that study?
About what?
On the cerebellum.
So in most of these, as is true for a lot of us cortex-focused people, in a lot of these studies, we don't quite get full cerebellar coverage, so we don't get much interesting to say about the cerebellum, which is a lost opportunity, for lack of a better way of saying it. Okay, so now I'm going to give you sort of some details on what we did to answer these questions, and as I show some pictures, I'll comment on a bit more of what other questions people added. So first, luck and specific data analysis, replication. So the first study had 3 volunteers. We saw the same results as in all 3. That gives us some confidence that there's something legit happening here. We've done published a follow-up study that did the same thing in 3 more subjects. That gives us more confidence. We also did several variants of the same analysis. We did different models. We did different clustering methods. And And anything where we said this shouldn't affect the results, it didn't affect the results. Anything we said, yeah, that might affect the results, like if we modeled all the hemodynamic responses with a single shape, we didn't see as much activity as if we were modeling it saying, find anything that looks significant. And that is what we showed. So essentially, this is sort of like kicking the tires in your data analysis. And there's always a fine line between trying a bunch of things in analysis till you get the result you want versus trying a bunch of things to make sure that that your result is robust to variation. Yes?
Did you vary the time window, like for each like neuronal pattern? Like did the 20 patterns arise if you only used a specific time window? Like what if you shortened it or?
So in this case, the main result was done sort of not assuming any model shape. So we had, so the task, every trial of the task was a minute long. So it's 20-second stimulus and 40 seconds rest. And there was a 2-second TR, so we have 30 time points. So we were looking for any shape of those 30 time points that, that was repeated 100 times in a significant way. So it was, so the most robust analysis was the one that did that wasn't making any assumption about the shape at all. So how do we rule out head motion? In general, when head motion is a big effect, we're going to see sort of some characteristic things. You're going to see maybe one side of the brain, the edges of the brain are going to have really robust signals, and we didn't see that at all. We also recorded head movement parameters, and they were really minimal. I forget how minimal they were on these 3 subjects, but I know so Ben in the front row, we're working on one of the follow-ups. Do you remember the numbers from earlier today? That how much?
The Euclidean norm of like the 6 motion parameters less than 1 for 95% of the runs.
Yes. So essentially, there was less than 1 millimeter degree of motion for 95% of the runs in 2 subjects. So those 2 subjects were phenomenally low movers. Then there's the blood flow dynamics, blood steal comment that was made earlier. This is hard to be definitive about. This is a hard one, but the spatial variance of the response shapes don't match what we'd expect blood steal to look like. So there's models of what blood steal would look like, and it's sort of an area the same signal would expand wider with more data, but instead we were seeing quite sharp boundaries where the signal would have quite different shapes. So like a transient wouldn't really be what you expect from blood steal. And a follow-up study, which I'm going to show on the next slide, also showed that there was widespread activation that changed depending on the task. So that's this slide here. And this also gets at that partial volume in question. So the original study was done with bigger voxels at 3 Tesla. The follow-up study was done at 7 Tesla with smaller voxels. And you can see this is the exact same this is one volunteer was done with the exact same task. And I'm not sure if you could see this, but you could see that once we have the smaller voxels, things are much more constrained to gray matter than they were in the initial study. So it sort of pushes us more that this really is a primarily gray matter effect. We also did the exact same in a second subject. They did it with just the checkerboard without the attention task. So we don't know if their mind's wandering or not, but we also don't know. But it also means that we don't have the secondary task with button pressing and letter number discrimination going on. And we see pretty much the same result. We do the same thing showing the same checkerboard, no task, on only a hemi field the whole time. And here we see a lot of the brain active, maybe less. Maybe if we collected 18 hours of data on this volunteer, we'd get the whole brain. But you see that this shows, this is not what we expect to see as a blood steal effect or a partial volume effect, because there's structure to this. It depends on what task we're doing. And at higher resolution, we see a much more focused on gray matter. So to summarize this case study, was this just a lucky finding? I don't think so. We've repeated this now in 8 subjects, 9 hours of data in 8 subjects. Is it specific analysis decisions? I think very unlikely, because like I said, we tried a lot. Is it head motion? Very likely. Very unlikely. Voxel size, partial volume? Probably not. There still might be some issues in how that interacts, but a good portion of this effect is probably not. This global blood flow dynamics and blood steal. It might be a factor, and it might affect exactly how that shape looks in certain places, but I don't think it explains the big picture. So there's clearly evidence there's something neural happening everywhere. If we're modeling it all perfectly, I don't know, but it's there. It's there. So I kind of went through this case study just to sort of give you all a sense of what it means to sort of go through one study and sort of have doubts and how you address those doubts. And as a final summary, fMRI really does help us understand the brain. Yay! Even though we measure an indirect signal, it can be quite specific. There are many ways to confuse artifacts with neural signals if you're not careful. I've hopefully given you some thoughts on how to be careful. And you want to think about your choices from the entire process. So why you're collecting data in a certain way, why you're analyzing data in a certain way. And most important, just look carefully at your data. The number of studies, actually, I think this is finally stopped. But for a long time, people were showing studies in like the orbital frontal cortex, which is like right, in a frontal inferior area of the brain, and they're showing all these findings, and someone says, did you look at your signal dropout in the area? And the person would say, what do you mean? And then they'd put up the raw data, and there's no data there. I think the field's finally been embarrassed. Probably about 10 years ago, the field got embarrassed enough that enough people realized that they have to look at the raw data, but that's like the most extreme example. But you really got to look at your raw data, you got to look at your time series, you got to look at sort of unthresholded statistical maps, to really get a sense of what's happening in your data. And that's about it. Does anyone have any questions? Great. Thank you for listening.